Right. So I don't entirely agree with Avi on that. I think there are value from parcellation, and I'll try and give my reasons why I think that. Yes. So first, I want to tell you guys a little bit about some of the differences in how we have done our neuroimaging processing to enable more accurate delineation of boundaries in the brain. Basically, how to acquire, pre-process, and analyze your data to get as accurate a parcellation as you can. I'll talk about the multimodal gradient-based semi-automated parcellation that you saw a preview of for one of the areas. And then a little bit about individual subjects using the same machine learning algorithm, actually, that Avi was already telling us about. So first, how should we be analyzing our data to maximize our ability to accurately parcellate things? And so we came up with this idea of, based on all of the work that's gone on in the Human Connectome project, where we've tried to really improve the acquisition and the analysis strategies and also the data sharing strategies, 7 main points that we call HCP-style neuroimaging. And the first is to get as much high-quality multimodal information on as many subjects as you can. Not always the easiest thing to do, but if the NIH gives you plenty of money, a big stack of money, do it. Use adequate spatial resolution to image the structures you're interested in, and I'll talk a little bit more about that in a second. When you're doing your pre-processing, you want to get any distortions out of your data. You want to get effects of motion out of your data, but you don't want to be blurring your data. You want to keep, if you acquire at high resolution, you don't want to lose that high resolution. It really helps to represent brain structures according to the natural geometry that they take. So for example, for the cerebral cortex, it's a surface-like structure. It has a thickness, but at the resolution of MRI that we currently have, it's basically best represented as a surface. Then when we want to compare across people, we want to actually get our brain areas lined up, not just line up the folds, because the areas in the folds may not always correlate with each other. And then when if we are going to choose to use a parcellation, let's use something that's, functionally and structurally relevant to the brain, not just a parcellation based on, say, the folding patterns. And then finally, we really need to be sharing more than just 3D stereotactic coordinates in our papers. We actually need to share the statistical maps, ideally the effect size maps, so that people can take the data and compare it directly with their own study to see if boundaries line up or they don't. So what kind of MRI data did we use? Well, in the past, as we've heard, parcellations have been defined on several modalities. Architecture, function, connectivity, and topography is a set of 4 that is particularly famous. And in the Human Connectome project, we actually were able to measure all 4 of these. So we measured architecture with T1 and T2 weighted MRI to make myelin maps and maps of cortical thickness. And we used fMRI for the other 3 properties. And so I won't really talk about diffusion. We heard a little bit about that. And we got 1,100 subjects, 4 hours of data. So what about this question of acquisition resolution that I was mentioning before? So the mean thickness here of the cortex is around 2.6 millimeters. And historically, people have often been acquiring their fMRI in this resolution. So that means a given imaging voxel may span across a sulcus, or it will contain large amounts of white matter or CSF, and not just the gray matter. But high resolution, like we acquired in the Human Connectome project, we're basically doing better than the average cortical thickness, so somewhere less than 2.6 millimeters. Now the minimum cortical thickness is all the way down at 1.6 millimeters. So here's where we acquired our 3T and our 7T data. And there are a couple of other useful reference points to think about when you're acquiring your images. So if you want to actually treat the cortex as more than a 2D sheet, if you want to think about its thickness, you're going to need to be at least on average 1.3 millimeter isotropic. It basically is just dividing this mean cortical thickness by 2. And if you want to have 2 voxels at least in every part of cortex, you're going to want to be at 0.8. And it actually turns out for your T1 and your T2 weighted images, getting them down to 0.8 really makes it easier to define surface models because, again, you avoid having partial voluming between the white matter and the CSF. You can get pure gray matter voxels. And then for reasons of data cleanup, if you can get your TR down a second or less, that really helps. So as I was saying, T1 and T2, 0.8 millimeter isotropic. Space and MP range work well, and you can achieve these resolutions with a 32-channel coil. Functional imaging, we did 2 millimeter isotropic, but if you can be less, at least than the mean cortical thickness, you're sort of getting most of cortex, at least a single gray matter voxel. And go fast if you can. And Avi didn't touch on this, but also in the NIL where he's based, this has been really shown to be really important, that to get reliable measures of functional connectivity, you need to get a fair amount of data. You can't just get away with 5 minutes of resting state data. You know, it's been shown that the slope going up of improved reliability is quite steep, up to about 30 minutes, and then even more, it's a little bit more shallow. Resting state is what's usually used, but other kinds of fMRI may work. But to achieve these goals of spatial and temporal resolution, you're really going to be using a multi-band sequence. And then, of course, because EPI data comes off of the scanner distorted, you have to be able to undistort it so you can map it onto your structural images. You need a field map for that. And everything I'm going to talk today about is going to require just these 4 images. So, as I said, we want to remove distortions, and so that all of our images represent the physical space of the subject. We want to eliminate the spatial effects of motion, make geometrical models of our major structures, cortical surfaces, and subcortical segmentation. You know, it's really common practice in brain imaging to blur your data, but if you look across other fields of science, like astronomy, you know, if they take a picture of the sky, they don't then smooth it a bunch and make it harder for them to see what's going on. So I really think that's not a good thing to do, and we didn't do that, and I think it really helped us. Then we need to get our areas aligned across subjects. Another big source of blurring in brain imaging is group average maps where areas aren't lined up across subjects. So how can we sort of represent the data to give us the best shot at doing that? And so one of the things to come out of the Human Connectome project was a different sort of analysis framework for representing fMRI data in particular so that we can achieve better brain alignment. And so we consider our gray matter structures according to the geometric model best suited for each. We'll use surfaces for the sheet-like cerebral cortex, volumes for our globular subcortical nuclei, and we'll use this concept gray ordinates here. So we have our 90,000 gray ordinates all on the end, and that's made up of 30,000 surface vertices for the left hemisphere, 30,000 for the right hemisphere, and then there are 30,000 subcortical voxels in these 10 different structures. And we'll be registering our data, the cortical data using surface registration, the subcortical data with volume-based registration. And just changing from the standard volume-based approach to this approach really allows you to improve your spatial localization ability and to reduce the amount of blurring. But also, how do we align cortical areas across brains? We'll take area MT as an example here and just compare what different registration algorithms can do. So this is, it's actually area HOC5, which is thought to be very similar to area MT. And you can see in a volume-based registration, we get these 2 hotspots of relatively low overlap. We're only maxing out at 46% overlap across 10 subjects. If we take the exact same data, but just go onto the surface here, we do quite a bit better, up to 57%, but we still have some of these areas that are not in this group pattern here. So if we use what I call aerial feature-based surface registration, we can drive that alignment up to 100%. And by aerial feature-based registration, I mean the same kinds of things that we would use in brain parcellation. So architecture, function, connectivity, and topography, or even just using the areas themselves. So this course, we want to tell you guys about some of the tools that are available to let you use some of these methods. So acquisition protocols and pulse sequences are available here, and these slides are all posted online, so don't worry about getting the links. Multiband pulse sequences are publicly available. We have a minimal preprocessing pipeline that we ran all of the HCP data through, and that's publicly available for you to use on your own studies. The MSML aerial feature-based surface alignment pipeline is also available, as is the registration software. to visualize data in this SIFTY format, we've released a Connectome workbench, the viewer, and also the command line tools. FSL is beginning to implement SIFTY, and certain tools like Melodic already will work with SIFTY natively now, or the film binary. And of course, you can analyze your data in MATLAB. Then we've released data from the Human Connectome project. It turns out 1,113 3T subjects and 184 7T subjects are available. And also the multimodal parcellation that used this data, it really wouldn't have been possible if we'd used traditional methods. We really had to move to these new methods to be able to do this. I'll put in a quick plug for the HCP course if you want to learn more about this topic. It just happened this year, but keep it in mind next year. All right, so multimodal gradient-based parcellation. I'll be the fifth person to answer the question why parcellate the cerebral cortex, but it's really important for what we're thinking about and what makes us uniquely human. And as others have said, it'll help us to understand the brain and communicate between investigators. And just as an illustration of how some of the both parcellation and some of the analysis methods I'm suggesting, this is a paper by Marty Serino from 1995 where he's mapped retinotopic fMRI onto the cortical surface. He didn't smooth it. His voxel resolution wasn't quite as good as ours is today, but still we're able to identify area V1, V2, and V3, and in his data from 1995, just like in Human Connectome project data now, we can identify these areas. And it's because he's not smoothed the data, and we've got a single subject, so we're not blurring across different brains here. So parcellation is often useful when you want to reduce the data size and complexity of complicated neuroimaging data while enhancing your statistical sensitivity and power. And you can think of it, there's a lot of smoothing in brain imaging, but if you're going to do smoothing, why not average within well-defined cortical areas? It's a neuroanatomically informed smoothing method. And for many types of studies that might put an MNI coordinate table somewhere in their results, if you parcellate the data first before, say, fitting a task GLM, and then just analyze the parcellated data, you'll have higher statistical sensitivity, you'll have better power because you're not doing as many statistical tests, and you actually have the areas already defined. So it's something to consider. What do we want in a cortical parcellation? I may have a somewhat different take on this than others out there, but there were 4 qualities that I thought were particularly important. It should be based on a lot of data, hundreds of really well-aligned subjects, so that what you're getting is really representative of the population that you've studied. You want to use the complementary and converging evidence available in multiple modalities, and ideally you want to cover the whole cortex. You want to make reference to the prior literature. Certain parts of the cortex have really been studied in detail, using histological methods and functional methods, and we want to, if somebody's already discovered an area, you want to, give them credit for that and use the same name. And then we want to be able to take this group parcellation, which may not exactly map onto individual subjects in exactly the same way, and we want to be able to find these areas in individual subjects. So we'll talk about all of this. What do I mean by hundreds of well-aligned subjects? Well, what I mean is, if we take a myelin map, which we can see here, we've got our red is our high myelin, and our blacks and purples are our low myelin. We've got, this is a group average of 210 subjects. We'll call it 210P, and we'll use this group throughout the talk here. This is another independent group of subjects, no family relationships between these two groups, no subjects the same. We've averaged on the surface after doing the aerial feature-based registration. We haven't done any smoothing. We're able to see some very fine details in the group average, like this little drop here in myelin content. That's actually right between the face somatosensory representation and the hand somatosensory representation. Turns out people have looked in monkeys, for example, done myelin stains in monkeys, and see exactly the same gap in between the face and the hand representations of sensory cortex. Same is true of fMRI. If you don't smooth and you line things up, you could see very fine features like this one here. It's actually part of area 55B in the right hemisphere. And the correlations between these maps are very high. So we're going to use this as the input for our parcellation, and we have a high expectation that if we make a parcellation based on this group, and then we validate it in this group, we would expect to see something very similar because the data that's being fed in is very highly correlated. So how do we turn this into a parcellation? Most of the extant parcellations out there are based on a single modality. It could be one of architecture, function, connectivity, and topography. But since we measured all four, we said, let's use all four. And as Avi told us about, gradients are a really useful way of objectively defining where a modality is changing across the cortical surface. And these are potential areal boundaries, and they're also a common framework that we can use to compare across different modalities. But what makes a gradient convincing as an areal boundary? Well, if we've got two independent modalities that are saying that there's a gradient in the same place, to me that's a really strong replication, and it makes me really believe that there's a real boundary there rather than it just being some kind of noise. We want to have statistically significant and robust differences across multiple modalities at the boundaries. If it's present in both hemispheres, it might not have to be present, but if it is, that's kind of an internal replication already. We don't want gradients that come from imaging artifacts. We have prior literature evidence. That gives us more confidence. And once we've decided on the set of boundaries that we want to use, then we want to relate these boundaries to the literature to see which of these areas have already been defined and which are new. So very quickly, here we have the myelin maps, but we want to find cortical areal borders, so we focus on where the myelin changes. Instead of being more high myelin, now we've got the reds, yellows, greens, and blues representing the where it changes rapidly, and it's these local maxima of these gradients that are most likely areal boundaries. We can do the same thing with cortical thickness. It doesn't have quite as much information as myelin does, but if we look over here in the central sulcus, we can see a very strong difference in cortical thickness from the posterior bank to the anterior bank. And so when we take the gradient, we see there's a strong gradient there. And that can be very helpful for telling sensory and motor cortex apart. You can do the same thing with task-based functional MRI. Here we have the subject is either listening to a story versus just resting in the scanner. And so the positive areas have more activity in the task, whereas the negative areas have more activity at rest. And you can use the same gradient-based approach on task fMRI data. I'll make one important note here. So here we use the effect size maps. So these are not p-values, they're not z-statistic maps, they're not dependent on the amount of data you have, they're not dependent on the field of the noise, they're just basically it's like a percent bold change map. And I think that's really what you would want to use for parcellation. You want your boundaries in the brain to be based on where is the actual biological effect changing, not necessarily where do the statistics change. Avi's already told you a little bit about this, functional connectivity from a seed location here. And we can step across resting state gradients and see the functional connectivity pattern changing. And some functional connectivity patterns look very similar between resting state and task, as Avi was telling us about. What about topographic organization? So this is a somewhat different concept than these sort of sharp gradients at areal borders. And by topographic gradients, I mean slowly varying, smoothly varying gradients within a cortical area. And often this occurs in cortical areas that represent, say, the visual field or body surface or sound frequency. So we have a map of the visual field, we have a map of the left hemisphere visual cortex, and different parts of visual cortex represent different parts of the visual field. So for example, if you were to stimulate here and shine a light in this part of the visual field and then measure the functional activity, you're going to get activation in this part of V1. Similarly, if we had V2 drawn out here, we get a little activation there in V2 and V3 and so on. And you can measure this either with a task paradigm or with resting state connectivity. And so these polar angle reversals for the visual field actually are really good for telling us where visual boundaries are. So we've got the horizontal meridian and the vertical meridians, and the first horizontal meridian runs right down the center of V1. The two vertical meridians tell us the boundaries between V1 and V2, as Professor Zillis was telling us. Between V2 and V3, we now have a horizontal meridian. And also these visual areas are representing eccentricity in one part versus another part. And so we can use resting state to measure this as well. And I'm just going to give you a very quick overview. It's a somewhat complicated method. So see the paper for a proper explanation. So we're going to take a map here, and this map represents a contrast in the resting state, but it's a connectivity contrast between the horizontal meridians, both of them, and the vertical meridians. We're a little washed out here, so I'm going to show down here, basically, this is the signal intensity profile. So it goes right along this line here, and you see it starts out positive, and then it goes negative, and it sort of oscillates. It's a little hard to see that, but if you actually take the gradient of this map, you can see that these oscillations. So now, right along the horizontal meridian, instead of the high gradients, we're going to focus on the low gradients here, basically where we're having this gradient go to 0, where we have a peak in this map. So when we're right along the horizontal meridian, the gradient is 0. Then we get along the vertical meridian, our gradient is going to be 0 again. So you can see these sort of minima. There's one right here. There's another one here that I can't see from this angle, but there are further ones going up that way. Now, if we take that gradient, instead of just looking at the magnitude, which we have here, we actually take that gradient and multiply it by another gradient that just points towards area V1 with a dot product here. Now we're going to get different signs in the visual areas. So V1 here is negative, V2 is negative, V3 is positive, and so on. So this is going to be important later when we want to use a machine learning algorithm that doesn't know anything about sort of this kind of a spatial pattern here, but it can interpret these signs flipping like this. So here are the early visual areas, and we could see they're heavily myelinated, and we're able to map their extent very nicely. So Avi stole this slide out of my talk, but he took the old slide. So this slide is actually much better than the one that he had, because it's got some new data cleanup methods in it that get rid of the red haze, we can say, right? So here we're going to focus on this slightly myelinated area here. It's right in front of the motor cortex, and it's got the two eye fields on either side of it. We've got our gradients, as Avi told us about, our task activity, our resting state functional connectivity. Well, the red haze is nowhere near as bad as it was when Avi had it. We've got a seed here in the posterior sylvian fissure, specifically connecting to this location that we saw is different in myelin and different in task, a different seed up here, specifically connecting to the either eye fields in our gradients, show a boundary that's lining up with the myelin and the task. Multiple independent modalities are telling us that there's a boundary here. To me, that's very convincing evidence. And then we want to look in the literature to see, has anybody seen something like this before? It turned out that guy Adolf Hopf, back in the 50s, he made some of the first sort of surface-based myelin maps. They were hand-drawn, but these are maps of myelin density. And so you can see there's a strip here corresponding to the motor cortex, and then two on either side here, regions of slightly heavier myelination than this one here. He called this area 50 by B, so I follow his lead. All right. There are topographic subdivisions in sensory and motor cortex. We can use myelin and cortical thickness to very nicely define the sensory motor areas, as has been understood with either histological mapping like cytoarchitecture or functional stimulation studies. But if you look at the boundaries found by functional connectivity or task activity, you see they're kind of orthogonal to those. So there are actually 5 well-defined subregions that go along the sensory motor cortex, but the boundaries are doing the opposite thing. So we've got our face down here. We didn't specifically test this in the HCP, but there's another study that shows that this seems to be particularly related to closing and opening your eyes. Basically, if you look at my face, see what I'm doing, those muscles. The hand representation here that was tested, we've got another untested, but likely the trunk representation, and then the foot up there. All right, so the multimodal parcellation, we constructed it from the 2010 P group of subjects. Borders were defined using gradients in the group average data. We used the architecture, function, connectivity, and topography, and we used a semi-automated parcellation approach, which basically the neuroanatomists, myself and David Van Essen, we looked at the different modalities and we saw, for a particular border, which ones seemed to be informative. And we drew an initial boundary where we thought the boundary was. And then we used an algorithm to take all of the information that we had chosen, multiple modalities, so it could be the resting state gradient, the task ephemeris, the myelin, and actually weight those together and pick a best path between them. And we identified areas with reference to the literature, keeping the names the same if possible. We had some predictions going into making this parcellation based on a human or a monkey parcellation and a partial human parcellation. We thought there would be somewhere between 150 and 200 cortical areas per hemisphere. There'd be wide variability in areal size and shape across the cortex. You look at some parcellations out there, and it looks like everything is sort of around the same size, sort of circular type things. But we didn't expect that. But there will be examples of interareal heterogeneity, for example, within sensory motor areas. So here are the borders that we found, 180 areas and complexes. We called something a complex if there was literature evidence that it could be subdivided, but in our data we couldn't see those subdivisions. Maybe our data wasn't good enough, so we'll say that maybe future data will be able to subdivide per hemisphere. Wide variability in areal size and shape across the cortex. And we're actually able to get the cortical areas in some places where the organization is well understood, but some of the other methods don't seem to do it. So for example, we get these strips in sensory motor cortex where we know, there's a particular architecture and particular function and computations that are going on, say, within motor cortex that are distinct from those on the other side in somatosensory cortex. Same is true in visual cortex. We know that different parts of visual cortex represent different parts of the visual field, and so functional connectivity, as Avi showed, is going to be stronger between, say, this part of V1 and that part of V2 than it's going to be between this part of V1 and that part. But we're able to define the whole area using this approach, V2, V3, and so on. No, I'm using the... Yeah, right. Topographic subareas, yes. So we could colorize this parcellation using basically trying to show where the different main input streams of the brain are going. So auditory, visual, and somatosensory motor streams for the colors, and then light and dark for the default mode task negative versus task positive. So one question, we made the parcellation with 2010p. If we look at a completely independent data set, do we actually find that there are robust and statistically significant differences across the areal boundaries? We're going to use 4 categories of features. So we'll use cortical thickness, myelin, task activation, and resting state connectivity. And our degrees of freedom here will be across subjects. We'll set a couple of thresholds here. So we want a robust effect size. We'll say Cohen's d greater than 1, which is really big effect size, actually. And then we'll be very conservative for our p-value. We'll do a Bonferroni correction across all of our tests and our 2 tails here. So we've got, of these feature categories here, 4 categories past these thresholds. Everywhere you see that it's red, and that's about a quarter of the boundaries. Orange is about half of them. Another quarter is 2. Only 40 boundaries passed those thresholds for 1, and 2 boundaries didn't pass for either, although it turns out if you actually get rid of the Cohen's d threshold and you just look at the p-value, even these ones that didn't pass the effect size passed for multiple for the p-value. We can also use this way of visualizing the boundaries of the cortical areas and coloring them in a certain way to see which modalities were useful for which areal pair boundary. So cortical thickness here we can see it's somewhat useful, but it turns out it's the least useful. It gives us the fewest yellow edges here. Myelin gives us more edges. Task ephemeris gives us even more, and a resting state is the most useful. So now we have a parcellation. What can we do with it? So here we have our myelin maps and we have our boundaries overlaid on the myelin maps. And this data you see here is about 60,000 data points, basically the 30,000 vertices in each hemisphere. We can average within each parcel like this and taking it down to 360 data points. So we go from 60,000 to 360. We lose a little bit of detail, but honestly not all that much. And if our parcellation is doing a good job, we're not going to lose too much detail. We can do some interesting parcellated analyses. We can make a functional connectome using full correlation with a seed here in area PGI, or partial correlation of the same seed focusing on the default mode network. We can do task fMRI analyses like I was mentioning before. Basically, we average the time series before fitting the task fMRI GLM. So here's one output group mixed effects contrast of that. And this is all of the HCP's task data. And this column here is what we've displayed here on the surface. Here's a different contrast. And we can compare this one actually to what we would get if we just did a typical analysis, a gray net-wise or voxel-wise analysis. And we can see that these are scaled the same way, so we can see in the map the statistical sensitivity improvements we get, and also in this graph if we just look across all contrasts. If they were the same, all of the dots would appear along the red line here, but you can see it's steeper, and this means that you gain statistical sensitivity if you parcellate first and then fit your GLM, versus if you do it the other way. You could fit your GLM and parcellate last. You do better if you parcellate first. So you can think of the HCP MRI data as a 3D matrix with parcels by features by subjects, a manageably sized high SNR data set that you can apply to your own data. So 180 cortical areas within each hemisphere within our expected range, vary widely in shape and size, and there's some heterogeneity. You can do a parcellated analysis with most modalities. We've got minimal loss of detail with a good parcellation. The group parcellation is available. This BALSA database is actually going to be a new database to enable people to share their results and not just the 3D coordinates. Connectome Workbench can help you compute gradients or even do the semi-automated approach that we used. And we'll talk a little bit about individual subjects now, parcellation of individual subjects. So we'll focus again on area 55B and how it can take different forms in different subjects. So most subjects have sort of this relationship between the upper limb sensory motor subregion and 55B. It kind of points down here. But some subjects actually have 55B up here. And so if we align the data based on aerial feature-based registration, this is the bigger feature, so this is what will get lined up, and this 55B will be misaligned. Now we could allow lots of distortion, allow it to tear the surface, and maybe we could shift this back down here, but that's not a great idea. So I'll show you what this looks like in real data. So here is the group average pattern. So this is a language network here. We've got our myelin map, a language task, and here's our upper limb region as our reference place. So an individual subject looks a little bit noisier, but if it's a typical subject, it fits what the group parcellation was showing. But there are some subjects that are shifted. So now instead of being within this group outline, we see that the connectivity and the activity is there, and now we've got high myelin here instead of low myelin. And we can see that in another shifted subject here. There's another pattern of 55B. It actually can split in half. So normally, 55B sits right in front of motor cortex and the two eye fields on either side of it here. But in some subjects, 55B gets split in half, and you've got the two eye fields coming together. So what does that look like? We've got most of the same maps as we had before, except now we'll have a dorsal stream visual network instead of the sensory motor subregion. Individual typical subject, we've got high connectivity and activity here. We've got light myelination with heavier on either side, and we've got connectivity to this dorsal stream on the two eye fields on either side. But here's the split subject. So now we've got two loci of activation and connectivity. We actually have this heavy myelination going right down this split here, and we have connectivity to the dorsal stream going right down the split. So we've got multiple modalities that are telling us that this subject is different from the typical pattern. See how they change together? There's another subject. It doesn't look exactly the same, but it's the same idea. We've got two hotspots of connectivity and activity. We've got myelin going right down the center, and along with this connectivity to the dorsal stream visual network. So what do we do about this? You know, if we're just we'll take our individual and register it, even with really accurate aerial feature-based registration to a group average, we're still not going to handle these mismatches. So we need to have a way of getting a match between them. Now, 89% of the subjects have the typical topology, but that means 11% aren't going to match for this cortical area. It turns out 4% have the shifted version I showed you, 6% the split version. We want the canonical parcellation to be based on the most common topology, but we've got to have a way of dealing with this. So we'll use the same multi-layer perceptron that Avi already told you about. We'll use the multimodal aerial features, so architecture, function, connectivity, and topography, instead of just functional connectivity, to learn each area's fingerprint. Make a few reasonable assumptions. We'll say V1 isn't outside of the occipital cortex, for example, or area 55B is within 30 millimeters in all directions on the surface of its group location. And we'll combine across our, we'll do a binary classification instead of trying to do all 180 areas at the same time, just to simplify things, and we can combine across classifiers later. So let's see, can we identify misaligned areas? So here we have some maps to see, and this is what comes the raw output of the classifier. We're able to find PEF, 55B, and frontal eye field, and after combining across classifiers. But this is a typical subject. What about the harder cases? Here's an atypical subject. We can see that 55B is shifted, and the classifier picks up on that. Similarly, if we've got a split subject, split 55B, the classifier picks up on that as well. So as I said earlier, some of the individual subjects were completely ray scanned through the HCP's protocol, so we can see how the classifier performs on 2 independent runs. So here we have those maps for a typical subject. We see folding, myelin, resting state look very similar, as do the parcellations. Shifted, or a split subject rather, we can see, again, the split in the original data and in both classifier outputs. So these shifts and splits are stable across time. These are real features of these subjects' brains. Across all areas, how do we do? Well, if we're yellow, that means we're detecting 100% of the areas across subjects. This is the data we trained the classifier on. This is data it never saw. We do very similar, 96.6% of areas in this map here of all areas across all subjects. If we leave out the task fMRI data, we actually do almost as well. So that means the classifier will work without the HCP's specialized task fMRI battery. T1, T2, field map, and fMRI will all work. We can make probabilistic maps of the cortical areas, and we see they look very similar between 2010P and 2010V. Some areas are better aligned with aerial feature-based registration than others. We already know 55B is not perfect. We're able to replicate the parcellation based on this aerial classification. So now we have our boundary gray ordinates shown, and so for 2010P, those are blue, 2010V are in red, and purple is an overlap, so these are well agreeing. Correlation is quite high, and this is, remember I said earlier, those dense maps we fed into the original parcellation had very high reproducibility, so we hope for this outcome, basically, and we got it. So individuals contain atypical layouts of cortical areas that cannot be aligned with aerial feature-based registration, but we're able to detect them with the classifier. We replicate the parcellation at the group level. We have these things to be available soon. Again, T1, T2, fMRI, and field map, and I'm only 3 minutes over. So I'll take questions. Thank you.
Questions, anybody? So I have a question.
Sure.
Because apparently Avi doesn't agree with us and doesn't think that parcellating the brain is moving forward. I think you stressed on a very important point by saying that parcellating the brain is going to improve the sensitivity, as well probably as the specificity of your statistical analysis. And if you want to give like some examples of future questions that we can assess now that we have good division of the brain.
Right, so parcellation is a tool like any other tool, like ICA is a tool, like functional connectivity as a tool, and it's not necessarily going to be appropriate for every question. So if you're, for example, really interested in the heterogeneity within cortical areas, it might be nice to know where the cortical area is, but then you might not want to do a parcellated analysis. You might want to say, okay, now within this area, can I understand, what's being represented? For example, we understand in visual cortex, there's a map of a visual space there. But what about in some of these more cognitive areas? Is there a mapping of some sort of a concept? It could be, language, for example, in language areas, or tools and tool use areas. Is there some sort of a mapping that is analogous to what we see in visual areas at this higher level resolution? And so a parcellation will be useful for telling you where the areas are, but you might not want to average across parcels. You might want to look at the finer detail within them. And as MR technology gets better, we're likely to become more and more interested in that fine scale level of detail. Yes, Avi.
Well, let me say that I agree with what you're saying. Obviously, parcellation has many clear uses. What I was trying to emphasize is what I think we should ask what it means that gradients are not comparably strong everywhere. Why does the brain choose to organize itself this way? That's the only point I wanted. It doesn't mean parcellation isn't useful, but that is a question which I don't see anybody asking.
I think it's an interesting question. That may depend on which modality you look at, for example. So say, you might be able to find a very weak gradient between the motor and the sensory half of the central sulcus in, say, task fMRI or resting state connectivity, but you find a really strong gradient in cortical thickness and in myelin. And so understanding why are some boundaries, between areas strong and in other boundaries much weaker, it probably will tell us something about, how, two neighboring cortical areas, what are they doing? Are they doing something very different from each other, or something that is very similar? Or it could even be very different. Motor cortex and sensory cortex have very different local circuitry. The cortex, if you look at them, histologically, they don't look anything alike. But in, functional MRI, because we're looking basically at correlations of the bold response, that bold response may be very similar between them. Whenever you use motor cortex, you must also use sensory cortex, for example, to know where your arm is in space, to move it. If you have your sensory cortex lesioned, you may not lose strength, but you'll have no coordination. It will be functionally useless, even if you still have the motor cortex. If you lose your ability to know where your arm is in space, you can't use it if you don't know that. So, some of that may have to do with the limitations of our techniques. Bold is a relatively indirect method. It's not telling us specifically what computations are being done in motor and sensory cortex. It's just telling us that they're active at the same time. So,
on a more general perspective, because you're expecting us to draw a hypothesis on why is the brain organized this way. Petrides and Pandya just published a book called Cerebral Cortex, where they provide an hypothesis about the organization of the brain in terms of sector architecture and connection. It's really interesting. I do recommend reading the book. It's really well written.
Cool. Yes.
What is your intuition about the computation that might be going on in 55B? Because you present evidence that it's the anterior primary motor cortex,
Right.
sandwiched between two gaze, eye movement areas.
Right.
Yet it shows task connectivity with the posterior superior temporal gyrus.
Right.
And it's very active during story listening.
Right. Yeah.
That makes my head spin.
Yeah, and then there's other studies out there. showing that it's particularly involved in other kinds of auditory tasks. So there's a study, and I'm forgetting the name of the guy right off the top of my head, but he's compared very similar tasks, except one is a visual and the other is an auditory task. And the visual task lights up the two eye fields, whereas the auditory task lights up 55B. You know, I would love to know if monkeys have a 55B. We don't really know that yet, but that might be a human-specific area that. You know, if you look across other tasks, though, even the HTTP task battery, there's this network of activations that are different in the left and right hemisphere. And 55B is one of them, where you have, you know, the language activation is much stronger in the left, but, you know, some visual spatial activations are stronger in the right hemisphere in the homologous location. And the same is true in basically other nodes of this network. So in the inferior frontal gyrus, where 44 and 45 are, actually there's a particular region, superior frontal cortex, that exhibits this behavior, where you look in tasks and you get these asymmetries left and right hemisphere. The same, that area at the back of the parasylvian fissure behaves the same way, and they're all connected in the resting state. And it turns out they also have one other unique property of connectivity in the resting state. Most cortical areas are most connected with their contralateral homologue in the other hemisphere. But for this network, that's not the case. It's actually the within-network connectivity that's the strongest. And so the left hemisphere areas are more connected to each other than they are connected to their homologues in the right hemisphere, and the same is true of the right. So this sort of asymmetric network is very it's very different from the way most of the cortex is laid out, and may have something to do why if you get lesions of the left hemisphere, you lose language ability often, and you get lesions of the right hemisphere and you lose visual spatial ability.
So if you use your 55B as a seed,
Yep.
standard
Right.
functional activity analysis, how does that map onto the big seven networks, big seven?
So it depends on who defined the seven. So in Carl Hacker's work, it maps onto his language network very well. Other networks, I don't think Yo has quite at the 7 level. He has a language network like this. With ICA, this comes out very easily as a particular thing. And if you do high enough dimensionality ICA, you'll get the left and you'll get very nice, you know, symmetric left and right hemisphere components that to come out independently. Yes.
I got a question about how to use the HSV-style neuroimaging for the clinical analysis. Now usually we use the normal parcellation to do the disease studies. When you got the schizophrenia, depression, using your HCB-style neuroimaging acquisition of the data sets, when you're using the individual parcellation, because their Maitland map changes, their connectivity changes, how to transfer your parcellation into the individual patients?
Right, so I did go through it very fast at the end there, but that was basically what our machine learning algorithm can do. So it learns in the, basically in one data set, the 2010P, what is the fingerprint of each cortical area? What is its myelin content, its function, its pattern of connectivity, its cortical thickness, and any topographic measures as well. And then when you give it that same information in another patient, you know, this classifier actually doesn't know anything about space. It just knows the pattern within the feature space, basically. What is the myelin, task, you know, connectivity, sort of write out in a vector for each spatial point, each gray ordinate. And so it learns what that fingerprint is in the one data set, and then you can give it new subjects, and it can tell you where those cortical areas are just based on the raw information that you're giving it. And it does a good job. It finds the atypical areas, and it's able to replicate the parcellation, and it detects the areas across most areas in most subjects, you know, 96.6%. So that could be used clinically. That would be an extension of what we already use clinically at Washington University for mapping neurosurgical patients. There we're just trying to get the functional networks, but we could try and take it down to the level of areas. We may need to bring MRI data acquisition up to a little bit higher level of quality, and also see to what extent, if we don't have the best quality data, can we still do some of these things. But that is for future work, sure.
And yeah.
Well, I had a related question about how robust it is on the non-HTTP data, but
Right, we don't know yet. That's something we have to find out. But I think it's going to have to come at both directions. In the one sense, we may be able to improve the method some, but the image quality is going to have to come up soon, some too. I don't think that 5 minutes of resting state data at low resolution or relatively low resolution structural images, you're going to really start to cut into what you can do for just those kind of regions I was showing before. Basically, the cortical thickness is so thick, and so you have to image at a certain resolution. And there are dynamics that Avi alluded to in the resting state. And so if you just get a very short period of time, you may capture a biased estimate of what the connectivity is, and you may need to capture enough time to make sure you're getting something reproducible. So these are important considerations. Yep.
Dare to speculate why the task-based studies did not help inform your parcellation procedure?
Well, they did. They were great for confirming boundaries, but it turned out that you didn't need them because basically you've got duplicate information, that the resting state in the task, at least in terms of telling you where boundaries are. The task is very useful because it could tell you something about function. Resting state can't tell you about that. But in terms of finding boundaries, they're duplicate. And there was a paper published around a little after this one in Science out of the Oxford group that showed that you can learn based on resting state data, you can learn a pattern of resting state networks and use that to synthesize task maps. So you learn that pattern in one set of subjects, and then you have only the resting state networks, and you can synthesize very nicely what the activation pattern would have looked like in the task data, and then compare it and show they do a very good job. So there's this, and this is, to some extent, but not as much true of even things like myelin. There's duplicate information here. So, for example, for the aerial feature-based registration, to show we weren't being circular, we held out the task fMRI data. We just used myelin in resting state, but we could dramatically improve the task fMRI alignment. So there's common information about where things are in the brain in these different modalities, and that's sort of what makes this work. I'm afraid I have to go catch a plane, but thank you.